I'm happy to be here today with Dr. Daniel Rhodes. He is co-founder and CEO of Compendia Bioscience in Ann Arbor, Michigan, and his talk was Target Discovery with Oncomine, Discovering New Targets and Positioning Old Targets by Integrative Analysis of Tens of Thousands of Tumor Genomes and Transcriptomes. Thanks for joining us.
My pleasure. Thank you.
Would you discuss your strategy for identifying novel targets and for generating new opportunities for existing targeted therapies?
Sure. So we really take an informatics approach, and we seek to bring together all of the published cancer genomic data that's been generated at various academic and pharmaceutical institutions. So when we bring together now tens of thousands of gene expression, DNA copy, and now somatic mutation profiles, we're able to take interesting analytical approaches to ask new questions of this large collection of data. And what we've done then is two things. One, we've asked, what new drug targets can we discover from this large collection of data by applying new analytical approaches? So to the DNA copy number, for example, we ask what minimal common regions of amplification occur in subsets of tumor population, and that leads us to new amplicons, often that are occurring in rare subpopulations that hadn't previously been discovered. So that's exciting. Similarly, on the somatic mutation data front, we employ sometimes very simple analytical approaches, but ask questions like, what hotspot mutations do we see in tumors occurring recurrently, so in several cases or small fractions of the population? This certainly identifies a number of the well-known cancer genes, but also sort of in the long tail of mutations that we're finding in this type of data, finding new recurrent hotspot mutations. So these both represent new classes of, albeit rare, driver cancer alterations that could be targeted. So that's sort of one aspect. Another aspect is we're taking this large collection of data and asking for the set of therapeutic targets that are already being targeted today, the targets that we already have agents for, at what frequency are they altered in human cancer? And what we're finding is much of what we already know, but also that these targets are implicated in tumors that they haven't previously been studied in.
And would you talk some more about that, what you found surprising?
Yeah, so I think probably the most exciting and surprising finding was that a number of very well-established cancer targets like HER2 and EGFR and BRAF, while their importance in their respective cancer populations is well established, so HER2 in breast cancer, EGFR in lung cancer, BRAF in melanoma, what we're finding is only after looking at thousands of patients that these targets are also altered, likely in a driver form, in rare subpopulations of a number of other solid tumors. So we might be talking about 1% of colorectal cancers or 1% of lung adenocarcinomas that have driver alterations in HER2. While this is a small subpopulation, this is still significant. This still amounts to thousands and in some cases tens of thousands of patients worldwide that have these alterations that could potentially benefit from therapies already on the market and approved for other indications.
So what are the next steps in this research?
Right, so from our perspective, we're an informatics company. We partner with pharmaceutical companies to help pursue some of the new targets coming out of our analysis. But for positioning existing targets and existing drugs, we're really now beginning to work with clinical collaborators at the University of Michigan and elsewhere to begin to consider large multi-arm clinical trials that are capable of targeting these rare subpopulations. Because if you think about 1%, you have to recruit 1,000 patients to get 10 that might benefit, and that's generally not been tractable. But if you can line up a whole number of these rare targets and give a patient a shot at, let's say, at a 10% chance of falling into one of 5 or 10 arms, now this becomes more interesting from a patient and a drug development perspective.
And in your opinion, what is the potential for genomic-based tumor characterization?
I mean, I think the potential is huge. There's a number of forces at play here, so I think it's just a matter of time before really all cancer patients are undergoing comprehensive genomic characterization, and that's being used to at first direct patients to really their optimal clinical trial. But looking further into the future, that will be used to direct patient to the optimal combination of therapies most likely to benefit their individual tumor. So the opportunity is huge, and it's a very exciting time to be in the field.
Thank you so much, Dr. Rhodes.
Yeah, my pleasure.